good morning. Thank you for being here so early on Sunday. So today, the title of my talk today is on the AHP Cannabis Monograph. I am on social media, and I'm also trying to just started this sort of cannabis science and history discussion group on Facebook. So if you're into discussing research articles and anthropology, ethnobotany, look for the group. So this is the monograph cover. You may have seen it. Like I said yesterday, I really love the cover. I think it's a great summary of where we are with medical cannabis, with the 10-foot tall barbed wire fence and the guard tower. But this book provides standards for basically, as it says, quality, purity, and potency of the plant. And I think this is really important. So being able to identify the plant, not just when you see marijuana growing, but how do you identify it in a product? How can we develop tests to get a finished, say, food product and know if it's made with leaf, flower, stem, root, and other products? This also helps provide standards for testing. So it has methods in there. It has some forward-thinking things. One of the things I really like about it is the information on cell culture, on how to sort of grow cannabis plants in little petri dishes. So we're going to talk a little bit about what the monograph is and why it exists. And so basically, to dumb it down a little bit, how does a plant become a medicine? And there's a sort of simple kind of formula is you write a monograph, then the herbal products industry writes standards, then you apply them together in some sort of certification or group. And so this is kind of what we have now is we have sort of nationally approved guidelines for the operators, for the laboratories, and cultivation is coming soon. And then we have this monograph, which is how to till the soil, how to grow the plant, how to test it. And this is some information that was talked about yesterday about herbal supplement acts and how they're used. So just in a nutshell, the Dietary Supplement Health and Education Act is basically that act that allows plant products to be sold at Whole Foods as herbal supplements. They just have to have a little warning on them about, this has not been approved by the FDA because the FDA does not research plant-based medicines. Oh, sorry, that last part's not in there, but the FDA, the FDA isn't really necessarily in the business of making plant-based medicines, but they are in the business of regulating them, which is a really kind of odd place to be. So we have to have monographs. We have to have a separate way to establish them as medicines in the guidelines. And so with the monograph and the APA guidelines, we can sort of start bringing standards. We have some fluid documents, the APA guidelines, which are, they're meant to change and adapt to things in the industry. And these are meant to kind of figure out, well, who's doing things right, This is not a strange thing for industry, for any herbal product, whether it's ginkgo biloba or sassafras root or whatever. People have standards, you have to deal with it. It's analyzed. If you want to see an example of what a natural products lab does, I'd recommend looking up Alchemist Labs in Costa Mesa, California. They screen plants using very simple techniques like thin layer chromatography, looking at contamination potency, and they use plant monographs in their study of cannabis. And so I'm not just here to plug the monograph book and to plug PFC, but I wanted to show you maybe some fun pictures from the monograph, my favorite sections, in hopes that it'll interest you in picking it up and reading it. It's only about 70, 80 pages long, and one of the first things it talks about is anatomy of the cannabis plant. So we have on the left, we have some very kind of historic uses, parts of the plant, different parts of the seeds, the calyxes, different types of morphology of leaves. There's a section about, sativa and indica phenotype, what those plants look like, but it's not necessarily helpful for looking at chemical profiles. But it does walk you through the anatomy of the plant. There are much more detailed pictures about the anatomy. For instance, one thing I learned while writing this is they're not called buds, they're called inflorescence. So that's a take-home message.
And it sounds much better.
Yeah, so those are some really nice inflorescence on the plant. But so it does talk about THC and CBD plants and have differences between them and discuss sort of the nature of these plants. This is one of my favorite parts of the slide. It talks about morphology of cannabis buds and inflorescence, and how they differ, what makes them that way, and sort of how you can tell what their growth characteristics were from the finished product. And the ones on the bottom left, I think, are the really important ones. And on the bottom left, you see two buds. One looks kind of smaller, maybe crispier, to use a non-scientific term. And I would just like to do a little interactive session. If you could guess what the difference between the buds on the left are and the buds on the right are, what would you say?
Drying. The worst ones are from New Jersey.
So, no, not New Jersey. Yeah. So drying. So if you say curing and drying, how old, and that is true, they are from the same plant, but how old are the inflorescence on the left? How old would you say those are?
3 months.
Let's see, those are a year old. So another thing that this monograph provides are storage conditions. So when you process something, how do you store it for maximum longevity, for maximum consistency? You know, we're not necessarily worried about making the most potent inflorescence you've ever seen in your life, but making a consistent, reliable product that can, you know, have a long shelf life. This is another one of my favorite parts of the monograph, and that's the discussion of trichomes. And I've had some really great trichome discussions in the last 48 hours. Scientifically. And so the important thing is one of my favorite things is High Times magazine. You know, they always put these pictures of like immature plants, immature fluorescence, and some of the pictures of trichomes that they show make me laugh because sometimes it's those bottom ones, the non-glandular wispy ones. And I think, well, there's not really much information you can get from trichomes that aren't producing cannabinoids except, you know, that they're not producing cannabinoids. But there are different parts of the plant produce different things. So depending, when I talked earlier about identifying a product, whether it's made from stalk, leaf, or inflorescence, that is actually important because the quality of the medicinal product, the quality of the cannabinoids will change. I believe that the sort of shorter, stockier ones that don't have the stalk with the cap, those ones are mostly like in leaf. And so if you're taking like outer leaves and making a product, that's going to be different than if you're taking something that has the typical stalk and cap trichomes. So this is a really kind of fun thing to learn about it. And if you don't really understand what trichomes are, they're these nice little waxy tubes that are like factories. And so the oil gets sort of manufactured and it comes out the top. And there's enzymes in there that synthesize, you know, things each step of the way. And it's mutations in those synthases that give us different varieties. You know, there's different enzymes. You know, the enzyme that synthesizes THC from its precursor, if that's knocked out or it's mutated, you're not going to have a big, strong THC producing plant. Anyway, so one of my favorite things is the trichomes discussion in the monograph. I think this is, you know, the cell culture of plants I think is really exciting. You know, I'm not fundamentally opposed to genetic modification. It's something we've been doing since the beginning of time. If you've seen the recent episodes of Cosmos, they have a discussion about dogs. That's a form of genetic modification. But I think the idea of being able to, if you have a strain and you want to preserve it for long periods of time without flowering it, how do you do it? This is one of the techniques at the University of Mississippi is to basically culture plants in a specific medium before transplanting them out into the field is one of the ways you can do it. And I think there's a lot of promise in this. I don't think they're really to the point where they can grind up the plants to their stem cells and mix them together and grow a fully functioning plant, but I think that's the direction it's going. And so to have a little bit forward thinking about how do you cultivate these plants in a petri dish, I think is no one else is having that discussion really right now. And you know, this isn't meant to put people out of business. It isn't meant to tell you what products you can and can't sell. This document was made with the help of people from all forms, all types of the industry. So we have, you know, people from pharmaceutical companies. We have Dr. Mahmoud El-Soli from University of Mississippi collaborated on it. Michelle Sexton was on there. But we also had people like Vincenzo De Marzo, who's, you know, one of the world's most published pharmacologists in the world. And we also had people who actually are in the medical marijuana industry, like Valerie Corral and Mike Corral. They have a process for processing their cannabis in fluorescence. And it's not just used to make a little bit, it's used to make a lot of standardized, reliable, consistent product. And so their method for processing cannabis is in this book. So the point is that these are not methods that are super high obstacles to achieve. These are methods that people are actually using in the field today, and it's people who are in collectives are able to do this. So I think these are really good guidelines. It's a bar, and it's a bar that people can pass. So it's not something to be intimidated about. It's something you can strive for. It also has some information about methods. So if you're interested in what an internal standard is, how do you what's the method you can use in your GC? Say you have a method that you've been working on. So you think you have a run that can analyze THC in 5 minutes. Maybe you could compare it to, you will compare it to this method to validate it. And then you should get the same number or a very, very close number. Or if you develop a machine, like say a black box thing, and you sprinkle weed on it and it spits out a number, maybe you could compare that to a test that's been used for 30 years by labs all around the world. Just maybe validate it and get rid of the black box approach. So I think that this will bring a lot of consistency, standards, and kind of demystify the testing process for a lot of people. Because if they can do the methods in the monograph and get the same results as whatever method they're using, then you know that it's validated and it works. So just to kind of sum it up a little bit, some of the things that this also has, like I said, it talks about potency, analytical testing, storage, has lists of pesticides, contamination, and also discusses microbial and fungal analysis and limits. I love this slide. This is not in the monograph, but I saw this at a presentation at NYU, and the researcher gave me the slide. And this is from GW Pharmaceuticals Grow House, and I thought it was a good idea of how to implement quality control. And all the little tiny boxes are HPLC machines, and you can see that between every process it's tested. So you start off with your THC dominant variety, your CBD dominant variety. It's cured, it's QC'd at that point, and then before it goes into liquid CO2, it's QC'd. After it comes out of liquid CO2, it's QC'd. Then after the peppermint flavoring is added to make it palatable, it's QC'd again. And then before it gets actually shipped out, it's tested again. So you have a lot of tests. And so, this is pharmaceutical grade level, but this is something that, could be used. Something to think about, food for thought about how these techniques can be applied and used to create a really standardized product. And so it also has lists of solvents and their dangers, solvents not to use for extracting cannabis. There's a big list in there and sort of a little rationale for each one. But just to summarize, the AHP, the monograph, people who made the monograph, they exist. Their existence is for advising governments about how to deal with plants. This is what they've been doing since in 1942 when cannabis and 100 other plants were removed from the U.S. Pharmacopoeia. They've been helping to reintroduce them since their foundation. And so APA has developed guidelines for the cannabis industry. And so you put these together, you get a certification process, you bring consistency and standardization to the business. So thank you very much for your time. These are some resources, apa.org, if you want to find the guidelines, herbal.hp.net, Safe Access Now, you should all know that website. And I do some blogging for East Coast resource sites at the Sensible Science blog at freedomisgreen.com. Thank you very much, and I'm happy to take your questions after the panel as well. Thank you.
Thank you.